Hello, everyone, and welcome to the OBBN 27 Taxa Benthos Identification Review through the Woodlands and Waterways EcoWatch, which is a program that is a part of the ULink Center for Community-Based Research based out of Haliburton, Ontario. Today, we'll be going over how to identify the 27 taxonomic groupings of benthos macroinvertebrates, a part of the OBBN or Ontario Benthos Biomonitoring Network. We'll start things off by going over some different terminology and orientation, and then we'll get into the key ID features of all of the 27 groupings. Here we go. So starting with orientation, these anatomical terms are used to describe the locations and positions of different key ID features of the OBBN taxonomic groupings. So to start things off, we have interior, which is anything that's located at the front of an invertebrate, such as antenna or eyes. The posterior is anything that's located at the back end. So for example, we have 3 posterior tails here. Dorsal is anything that's located on the top side of an invertebrate. Ventral is anything located on the bottom or underside. And then lateral is anything that's located on sides. So for example, lateral gills are present here, you can see. Moving on, in terms of morphology of invertebrates, many terms you're probably familiar with, such as head or eye or antenna, but there may be some that are a little bit foreign to you as well. So to start things off, the head is always where the eyes and the antenna will be located if those body parts are visible on an invertebrate. We also have invertebrates that have legs visible on them. Sometimes these legs are segmented. So you can see we've got one segment here and a second here. And sometimes legs will terminate in tarsal claws as well, which you can see here. You can see this one has one, but some invertebrates may have 2 tarsal claws. So that makes it a helpful feature when trying to differentiate between different individuals. We also have an abdomen, which is made up of segments. So you can see this one has 1, 2, 3, 4, 5, 6, 7 abdominal segments. Sometimes the gills are present on the abdomen as well. Sometimes they're present at the end of it, or at the end of an abdomen. But we also have tail here at the bottom of, or the posterior end of the abdomen. So that's another feature. Some other additional terms, I already mentioned terminate or terminal. So we can have terminal tails or legs that terminate in a tarsal claw. Also have the term sclerotized, which essentially just means hardened. So some invertebrates will have sclerotized body segments or portions of their body. Another common term is proleg, which is essentially a leg that's underdeveloped. So it's smaller and usually is not segmented. So now we'll move on to our first invertebrate grouping. So the Coleanterata or hydras are essentially the 2 with tentacles. That's what it looks like here. They're inconspicuous, which means they're difficult to see. They're very small, 2 to 25 millimeters in length, and they can vary in color, but they're usually clear white in color. They're sessile, which means immobile. They sort of adhere themselves to different things in rivers and flowing water, so potentially a stick or something like that, and then they'll just hang on to that. The turbellaria or flatworms, so they look like what they sound like. They're flatworms. So they have eye spots that are held dorsally on the head, you can see here, and the head is usually triangular in shape. They have a mouth that's held ventrally on the head, and they sometimes have a pharynx, which comes out of the mouth, which is just a tube that sort of looks like this, and it's what they'll use to suck up food. They are 5 to 20 millimeters long, dark in color, usually gray or brown. And they don't swim, but sometimes you can see them creeping along the bottom of the tray or dish that you're using to pick and tally your bugs. Next, we have nematoda or roundworms. These are unsegmented worms. They're often clear in color. They're less than 1 centimeter long, and sometimes you can see the innards within them. So in the individual at the bottom of the screen, you can sort of see the coloration of the innards in exhibit there. Again, if you are live picking, you can see them moving rapidly in a whip-like fashion along the bottom of the tray, and their head is tapered and the tail is very pointed. Okay, oligo or aquatic earthworms. They are 1 to 30 millimeters long. They have a segmented body, which you can see here, different segments. And at each segment, they'll have thin bundles of hair. They look just like a terrestrial earthworm, but smaller. They have no eyes or suckers, so they can't be confused with Turbellaria or Hyradinia, which we'll look at next. And they're usually coiled when you're picking for them, but they can also be seen crawling along the bottom of the tray as well. So now, Hyradinia or leeches, another worm-like individual, but they do have suckers at both ends of their body, at their anterior and their posterior end. So you can see the sucker here, and this is the sucker here as well. They move along by inching along the bottom of the tray as well. They're usually 5 millimeters to 30 centimeters long, so they can get very large. And they do have several pairs of eyes, so a little bit difficult to see. They do have eyes down here, and you can see the 2 dots there for their eyes right here. In color as well, they can be brown, olive, black, or gray. Now we'll take a look at the isopoda or sow bugs. They are essentially the aquatic version of roly-polies or potato bugs, whatever you know those little terrestrial bugs as. So they're like little armadillos. They're compressed dorsoventrally, so from the top and from the bottom. And they have 7 pairs of legs on their abdomen that they'll use for crawling, and the first pair of legs does have claws present on them. They have 2 sets of antennae, with the first always being longer than the second. For our bivalvia, which are clams and mussels, they have hard oval shells that are hinged and have an opening. So you can see the hinge here and here, and of course our opening. They can range between 2 to 250 millimeters in length, and they have rings that you can see present on their shell as well. A very important thing to note is that you do not count empty shells, because that means there's no invertebrate actually present within them. Next, we have our amphipoda or scutes. So these are compressed the opposite way as our isopods or isopoda. They're compressed laterally from the sides, and they essentially just look like little shrimps. They have 2 pairs of long antennae, and they have 7 pairs of walking legs. They have 6 segments on their abdomen, and they're 5 to 20 millimeters long. They're variable in color as well, so they can be pink or white or gray, brown. Sometimes they're also translucent, so you can sort of see through this individual under the microscope. Next, we have our Decapoda or crayfish, which are essentially small little lobsters. They have a cylindrical front or anterior, part of their body, and they have a dorsoventrally flattened posterior here. So this is very flat. They have 7 pairs of walking legs, and the first 3 have claws, with the first having very enlarged claws, as you can see here. They have very hard shells and their eyes are held on stalks. They have a broad telson, which looks like a tail, but it's just the sort of last segment of their abdomen. And they'll use that for swimming backwards. So you may see that when you're out sampling for these guys. They are usually 1 to 15 centimeters long. And again, they can vary in color: blue, green, brown, and sometimes red or rusty. Next, we have our hydrachidna or cnida and or mites. So we have adults with 4 pairs of segmented legs, which you can see here, 1, 2, 3, 4. They are circular or sphere shaped. They do have eye spots, so you can see those here and here, and they're usually brightly colored. So you can see this one's blue here, this one's sort of got blue and orange in it, but they can be red or brown as well. They really just look like small spiders or ticks. So they're usually 1 to 7 millimeters long. Next, we have our Ephemeroptera or mayfly. So they have 3 tails, which you can see here, but sometimes their tails can be missing, so you can't always rely on that. They have 1 single tarsal claw at the end of each of their 3 sets of legs. They also have visible gills that are always held dorsolaterally on the abdomen. So they can appear plate-like, like this individual, or they can appear feather-like, such as this individual and this individual here. They are usually 3 to 28 millimeters long, but that does not include their tails. Next, we have Anisoptera or dragonflies. So you'll notice that these individuals have wing pads that are present dorsally. So they might be a little bit difficult to see, but they are held here on their dorsal side. They also have a modified labium on the dorsal side, or the ventral side of their head, which extends for catching prey. So this is their labium here. They're larger than damselflies or Zygoptera, which we'll look at next, because they have a bigger head and bigger eyes. And they have just a larger body overall. They have no visible gills. They appear flattened. They're usually 15 to 45 millimeters long, and they actually have jet propulsion from their posterior end. And usually they're green or brown or dark in color. So next, we're taking a look at our Zygoptera or dragonfly. So you can see here that they're much more slender than the dragonfly, or sorry, we're looking at the damselfly, but they are more slender than the dragonfly. So they have wing pads present as well on their dorsal side. They do have a modified labium for catching prey as well. They actually have 3 distinct gills that are held at the posterior end of their abdomen. So they look like tails, but they're actually gills. And they're usually between 1 and 22 millimeters long. And they're usually green, beige, or brown in color. Next, we have our Plectroptera or stonefly. These individuals do resemble Ephemeroptera or mayflies, but the key difference is that they have only 2 tails. So you can see that here, 1 and 2. Their tails are held ventrally on the abdomen, so they're always underneath. You'll never see them on the side. And sometimes they're also present on the thorax or head. They also have 2 tarsal claws, which you can see here, 1, 2. Opposed to the single tarsal claw that Ephemeroptera have. Usually they're yellow, brown, or black in color, and they do actually require fast-moving water in their habitats. So you generally won't see them in a lake, but mainly just in rivers. Moving on, we have hemiptera or true bugs. They are 15 to 40 millimeters long and have sucking mouth parts that are similar to a beak. So we'll use that to pierce their. They do not have gills. They actually breathe air, and they have 2 claws on some legs. They usually have 2 pairs of membranous wings that overlap, which you can see here. They're overlapping. Next, we have Megaloptera, which are fish flies, alder flies, or Dobson flies. These individuals are 25 to 90 millimeters long. They have 3 pairs of legs that terminate in 2 tarsal claws. They have lateral abdominal gills. They have well-developed mandibles, which you can see in this image, and you can see down here as well. They have a sclerotized head and thorax, which again means that it's hardened, and the rest of the body is soft. Depending on the family of organisms, they will terminate in, their abdomen will terminate in 1 long segment, which you can see here, but additional families or families that are different than the one pictured here will end with 2 anal prolegs or hooks. Next, we have Trichoptera or caddisflies. These individuals are 2 to 50 millimeters long. They have 3 pairs of legs, which you can see here and here as well, which end in a single tarsal claw, and they have 2 anal prolegs, which you can see here as well as here. Will sometimes be inside a casing that's made of woody debris or rocks and sand, and 1 to 3 of their thoracic segments, so these segments that are a part of their thorax, may be sclerotized. So you can see we've got 1 of them sclerotized here, and these 3 are sclerotized. It's important to note as well that if you encounter a casing that does not have anything in it, such as this one here, you do not count it. Next, we have our Lepidoptera or aquatic moths. So you can see they've got 3 pairs of segmented legs that are very short, located on the thorax here. And they do have abdominal prolegs. Remember, those are undeveloped small legs that end in tiny hooks. You can see them here, the prolegs. They are 10 to 25 millimeters long. Will crawl just like a caterpillar. And sometimes they'll have filamentous gills that you can see on the body here. We have Coleoptera or beetles. These individuals are 2 to 12 millimeters long. They have 3 pairs of legs on the thorax. You can see them here, as well as here, 3 pairs of legs, and antenna with 11 or less segments. Adults will have wings that do not overlap, so do not mistaken them with true bugs. And the forewing, which is the hardened wing that you can see, called elytra. They'll have those present as well, not overlapping. And larvae will always have a sclerotized head with mandibles and no wing pads. Next, we have gastropoda, which are snails and limpets. These individuals are 2 to 70 millimeters long, and they look just like terrestrial snails, except they're found in the water. So they have a hard spiral or cap-shaped shell. If you can see their bodies present, they have a prominent head that has tentacles. They may also have an operculum present, which is a hard flat sheet that's almost like a trap door that covers the opening. of the shell. But remember, if there's nothing inside of the gastropod or snail, you do not count it towards your tally. Next, we've got our chironomidae, or midges. So these are 2 to 3 millimeters long, usually white or red or greenish yellow. Sometimes they can be translucent as well. They have a well-developed head, which you can see here, that is sclerotized and does have eyes present. They have posterior parapods, which have hooks, which are essentially just small protrusions that you can see here at the posterior end. They also have an anterior proleg, which you can see here, which is held centrally behind the head. They're usually J-shaped, and they may also be found in a tube of dirt or plant material. They can also be caught in the surface tension of water when you're picking. So if you introduce a little dollop of dish soap, that will actually help to dissipate the surface tension when picking. Next, we have tabanidae, which are horse flies and deer flies. They have 3 to 4 pairs of creeping welts on the body. You can see here. Have hooks on each of the first 7 abdominal segments. They are pointed or tapered at both ends and have a leathery texture. They're usually 15 to 40 millimeters long, and their head is retracted into the thorax, so it's invisible. You can see that there. Next, we have Culicidae, or mosquitoes. They are 3 to 15 millimeters long. They have fused thoracic segments, which make their thorax visibly wider than the abdomen. They have hairs on the front of the head and sides of the mouth, and they have a respiratory siphon at the posterior end, which you can see here. And my pen won't show up, but here. Next, we're taking a look at the Ceratophragonidae, which are no-see-ums or biting midges. They are 2 to 13 millimeters long, very slender and pointed at both ends. They have a well-developed head, which is sclerotized with eye spots. They do not have a proleg or any appendages like the chironomid has. They may have terminal tuft of hair at the posterior end of the abdomen, but they are also smooth and shiny, usually creamy or white colored, and they generally remain stiff when you pick them up with forceps or tweezers. Next, we have the Typhoelidae, or crane flies. These are 10 to 50 millimeters long, usually white, yellow, or brown in color. They have a reduced head, as you can see here, which is retracted into the thorax. Their body is membranous and may have creeping welts present on it. They do have a respiratory disc at the posterior end with lobes, as you can see here. Next, we have the Simulidae, or black fly. This grouping is 3 to 15 millimeters long, usually brown or gray in color. They will inch along the bottom of the tray. They are flattened with their posterior end 1/3 fatter or larger than the rest of the body, as you can see here. They have anterior labral fans, which you can see here. And they're also sessile or stationary, similar to the hydra, which was the first grouping we looked at. So they actually have a suction cup organ at the posterior end of their body that they use to hold themselves to rocks and debris in the water. Lastly, we have the miscellaneous Diptera, which is any other true fly that is not represented in the 27 groupings. So are or maggot-like, Some may or may not have a head present, reduced, or retracted into the thorax. They will not have any segmented legs or any wing pads. They may have creeping welts or prolegs or terminal processes like respiratory tubes. So that is all of our 27 taxonomic groupings. So now we'll just go over some invertebrates that are not accepted by OBBN. So larval excavae, which is pictured here, are not counted towards your tallies. Again, like we've mentioned throughout this session, empty shells for clams and snails are not counted, and empty caddisfly cases or trichoptera cases are not counted. Individuals that do not have a head also do not count because we do not want to double count an individual by accident, as well as invertebrates that are missing too many body parts that they cannot be identified accurately. We also have some invertebrate lookalikes out there that you should watch out for. So some plant matter that you'll collect in your samples may look very similar to some different taxonomic groupings of invertebrates. For example, here we have a seed that looks an awful lot like a gastropod or a snail. It has that spiral appearance, but it does not have an opening, and it contains a seed inside. Sometimes you'll see grass or leaves in your samples that look like nematodes or another type of worm, but they're going to be missing those key features like the tapered and pointed ends. And as you can see, this one doesn't really have a translucent appearance. You'll also have non-OBBN invertebrates or zooplankton or phytoplankton collected in your samples sometimes. So make sure you're not counting individuals that are zooplankton, such as copepod or daphnia, or other types of zooplankton, as well as on OBBN invertebrates. So these are bugs or invertebrates that are present in the water, but they don't exhibit any of those OBBN taxonomic grouping features. So if they, you can't match them to any of the 27 groupings, chances are it's a non-OBBN invertebrate. That brings us to the end of our session. Thank you everyone for watching. I hope this was helpful for your research into OBBN. And if you want any more information about the Woodlands and Waterways EcoWatch or ULinks, you can head over to the link that is included here, which is just www.ulinks.ca. Thank you everyone for watching.